first of all, I welcome you all for this talk, Super Resolution Microscopy, which leads to technological advances to human disease. And I must tell you that we are very fortunate to have Professor Samuel Hess from University of Maine for the lab visit talk series. And of course, learning from the inventor himself is a great opportunity for all of us. And I'm very happy that he kindly agreed to give this talk. And for beginners, this is a great way to start their career. So I hope to hear his version to the path that led to the invention of super-res microscopy. And what I remember and what I have seen from that, he's the first ever scientist who has used super resolution microscopy to understand disease biology. And and I think this has led to the fundamental contribution to this field, to the whole field of biology, and specifically disease biology. So we'll just go ahead and hear from him. Sam, can you please take over?
Thank you, Partha, and it's a great pleasure to have a chance to talk with you and to present some of our research. Also, I would point out that this background that's behind me there is UMaine, but it's actually a picture taken when Partha was with me. So we have a strong connection for a long time, and I'm very grateful for that. So if I can go ahead, I will talk about some developments in microscopy. And I would also encourage everyone to ask questions. I really do this for interaction. And so please feel free to ask questions as I go or at the end, as whatever you like. So thanks very much for the kind introduction, Partha. And some of this work has been funded by National Institutes of Health, which I'm very grateful. I'm going to start with an introduction about the limits of microscopy and also the reasons that we pushed very hard to do better, to try to create new technology that could answer some fundamental questions. And I think that's sort of a philosophy that went along through my career, that there was a need for some kind of new information, and we were lucky to find ways to advance the technology to allow us to address those questions in biology, which then often led to new questions. So kind of step by step together. And the fundamental question really that's been on my mind for almost 20 years now has been the problem of influenza virus, which causes still significant morbidity and mortality worldwide. We have these great vaccines, but they have to keep being adapted to seasonal mutations, and some strains are resistant to the available drugs. So we have this pressure to try to better understand how viruses infect. Obviously, they can disrupt our lives dramatically, and yet they have to get in. They have to get into our cells through entry to be able to infect, and that entry depends on a process called membrane fusion, at least for enveloped viruses, where the viral membrane has to merge with the cellular membrane to allow viral genetic material to enter. And that membrane fusion step for flu depends on the clustering of a protein called hemagglutinin, or HA, which is the fusion protein or spike protein, as it's known in the case of SARS-CoV-2. This hemagglutinin needs to form clusters in the membrane of the virus to cooperatively open the fusion pore. And so the mechanism of clustering is an important place to attack the virus, and yet the mechanism of clustering is not understood. And so this is what we've been focusing on, trying to understand how clustering within the plane of the membrane of hemagglutinin occurs and allows fusion to proceed. And this is some pictures of the some pictures of the structure of the virus as it proceeds from its pre-fusion to fusion active or post-fusion state. You can see these trimers of hemagglutinin on the surface of the virus. So where is this in the life cycle? It's as the virus binds and is enveloped by the host cell endosomal membrane, the hemagglutinins undergo a conformational change that opens a fusion pore that allows the RNA to get in.
Sorry, Sam, just to interrupt you, your
Yes.
PPT is not shared. Yeah,
Okay.
now it's visible. It's quite clear. Yeah.
Oh,
Please
very
go.
good. Yeah, so sorry for the trouble. Thanks very much, Partha, for the chance to present some work about our developments in microscopy. And I'd like to acknowledge support from NIH and IGMS specifically. And I'm going to try to summarize my motivation for developing this new direction in microscopy, as well as how this happened, and then try to follow how the technical advances and the biological questions have been connected together through this process. So the problem that I've been focused on recently is the problem of influenza infection, which is still a significant trouble in the world, and of course connects with other respiratory viruses as well, SARS-CoV-2, for example. We have good vaccines, but because of mutations, the viruses need to be, the vaccines need to be updated quite frequently. And the entry of the virus depends on membrane fusion, which is the merger of the viral membrane with the host cell membrane, which depends on clustering of the fusion protein hemagglutinin, or HA. Yet the mechanism of HA clustering is not known. And so we've been focusing on understanding this mechanism of clustering, which could provide antiviral strategies that we can exploit. And this is in the, so where is HA playing a role? Where is the clustering playing a role? It's helping the virus to stick or bind to the host cell surface. Then endocytosis brings the virus into an endosome where HA undergoes a conformational change that leads to membrane fusion, which then releases the viral RNA into the cell, and then that cell is infected. HA must also assemble with other viral components at the cell surface to form new progeny. And so clustering may occur at this stage and plays a role at this stage and this stage of the life cycle. And we can study this with microscopy using a cell-cell fusion model where red blood cells that have the right receptor on their surface are labeled with a fluorophore inside, soluble dye, and a membrane, red fluorescent membrane dye on the outside, and are placed in close proximity to HA-containing cells, which can then be induced to cause fusion. And then by standard microscopy, we see dye transfer when that happens. The red and green dyes are transferred into the HA-containing cells, kind of like a virus infection model. Yet, this is conventional microscopy and has very limited resolution of the HA clusters themselves. You can kind of barely see heterogeneity on the edge of the cell, and in this case, very little heterogeneity. In this case, you can see some clusters, but many of them are too small to resolve. So conventional fluorescence microscopy doesn't have the resolution that's needed to see what's going on at the molecular scale. And this was a big motivating factor when I was a postdoc at NIH and learning about flu, that we needed better kinds of microscopy. And of course, the electron microscope has the ultimate resolution. This is some immunogold labeling of hemagglutinin on plasma membrane sheets. And these black dots show the locations of HA trimers, which are very much clustered. However, this is in a fixed cell far removed from living native membrane. So we still need something that can work in a living cell, which is going to depend a bit on some things that I did as a graduate student. So when I was a graduate student, the green fluorescent protein was becoming very popular, and the X-ray crystal structure had just come out, showing the chromophore could undergo transitions between blue dim fluorescent version called the neutral form with a proton on the chromophore, and the anionic form, which is green fluorescent, and the proton has moved to another location nearby. And W. Murner showed interconversion of individual GFP molecules between bright and dark states using different wavelengths of light. This was very intriguing to me. And I could see that studying the photophysics of GFPs could be very useful. We use a technique called FCS, fluorescence correlation spectroscopy, which is invented by my advisor, Watt Webb, where individual molecules diffuse through a diffraction-limited focus. Those molecules enter the volume, causing fluctuations in the fluorescent signal. You can autocorrelate this signal with itself and get a correlation curve, which shows the time of diffusion, the time the molecules spend within the observation volume, and also any transitions between bright and dark states that occur while they're in the volume, which appear in this part of the curve. And this allows quantification of the time molecules spend in a dark state, as well as the fraction that are in the dark state at a given time. With this technique, we were able to find that the populations of different states, neutral, ionic, and a second dark state, which is pH dependent, were strongly light dependent, and one could push the molecules out of one state and into a complementary state using different intensities of different wavelengths, 405 nanometers, for example, and 475 nanometers for the second wavelength. This I kind of tucked away in my mind after we published it, and then kind of went back to other things. And, you know, this photophysics didn't really lead to a discovery in itself. We still had the diffraction limited resolution of the microscope, right? You image a point source with a lens system, and you get a point spread function, which is approximately 250 nanometers in diameter, or given by this Rayleigh criterion, 0.61 times the wavelength divided by the numerical aperture. And that's much bigger than an individual hemagglutinin or other protein would be. So how do we get around this diffraction limit in the resolution? And this came back to the photophysics when I thought about trying to image small numbers of molecules that were well separated from each other. That's called localization. And a friend of mine, Russ Thompson, and another friend, Dan Larson, were working on this localization paper in the web group at the same time I was there, and saw that molecules could be found and localized with an error squared given by this expression, meaning that if the molecule is well separated from others, you can use the image of the molecule to find its location, plus or minus this sigma x, which depends on the standard deviation of the point spread function, the number of photons detected, the pixel size in the camera, and the background noise B. And so if we consider the diffraction limited resolution of about 240 nanometers, and we work on reducing background and making the pixel size appropriate for the camera, we can get this error to reduce to the standard deviation of the point spread function over the square root of the number of detected photons, where this number of detected photons could be close to 1,000, which would give localization precision of a few nanometers. So we could find where these molecules are plus or minus a few nanometers as long as they're well separated. That doesn't give us an image because it only provides a few localizations. If they're too close together in a system where we have many molecules, then this whole process will break down. So the use of the photophysics was the way of getting out of this problem. Instead of looking at the molecules all at once, we use photophysics to convert them all into a dark state or have them in a dark state to begin with. We use an activation beam, the 405 nanometer, to turn on stochastically a small subset of molecules to become fluorescent, which are then imaged with a second beam called the readout beam. And from those images, the positions are determined, and the molecules are then bleached, and another subset is processed in the same way. And we accumulate more and more subsets by acquiring more and more camera frames of different molecules until we can reconstruct the structure of the sample, but now not in a way that's limited by diffraction only, but rather limited by the localization error and the number of individual localizations, or the density of localizations within the structure we care about. So the data looks like a stream of spots, bright flashes of light from each molecule. The boxes are drawn to show the things that we identify. We then localize those things, plot a dot for each one. Sometimes we use the localization error as the size of the dot. Sometimes the mean localization error for the whole data set is used. But anyway, we plot each molecule as a dot and gradually kind of fill in like a pointillist painting, like a Monet or something, with each molecule being a single brush stroke and each brush stroke having a diameter equal to the localization error. And that then gives a structure which can be compared to the conventional image, and you can see much more detail. So going back now to what our goal was, to study how the flu protein hemagglutinin is organized, we see that in the conventional microscope image, much of the structure is blurred. And in the super resolution image, these clusters at the edge of the cell become much better visualized. And just to point out, this is not a complex system. It's a fluorescence microscope with a sensitive camera and 2 lasers. That's really the essence of it. So this is an accessible thing to many labs. And at the University of Maine, of course, we're not the wealthiest university in the world, and the ability to do this in our lab and develop it was really very helpful, and also not too expensive. This is one of our scopes. So we need fluorophores that can be controlled. We need photophysical control over the states. And a conventional fluorophore is not exactly ideal. It would be typically fluorescent. All of them would be fluorescent or potentially fluorescent until bleaching. And what's better is something which is initially dark and can be converted into a bright form by a separate wavelength, such that we have control over how many localizations are occurring per frame. Otherwise, it's going to get to be too dense, and the localization process may break down, or you need more complicated multi-emitter fitting algorithms. We could use something which goes from dark to bright, or something which goes from one spectral region to another. For example, photoswitchable protein, or photoconvertible, as some people call it, which starts with a green spectrum and is then converted to a form with an orange emission. And thus, in the orange channel, goes from dark to bright, but in the green channel is visible for most of the time and can be imaged with conventional methods. There are a variety of different kinds of fluorophores. I talked about fluorescent proteins. Those were some of the first used, and they have this wonderful genetic control. You can attach them genetically to a protein of interest. But organic dyes have advantages too. Large numbers of detected photons, smaller size in comparison to an FP, ability to conjugate directly to a molecule of interest. And then within those organic dyes, you can use photoactivatable or caged versions, or redox buffer can be used, and conventional fluorophores can be used. So what can we learn about influenza using these better views? So, right, the clusters here, which we said we thought were important, are much better visualized. The shapes are clearly not entirely rounded, which was one model. That's not the case. They're highly irregular. And why are they so irregular in shape? Looking in live cells, we could see something intriguing. And I would mention that we were the first to do localization microscopy in live cells in this 2007 paper, where we saw that HA was confined to these elongated regions and doesn't go into some regions of the membrane, even though there's plenty of mobility and time for the molecules to reach those locations. Why did they not go in some places? And why are the places they go elongated in shape? We thought at first that maybe this was just a blurred image of a vesicle moving over time. But when we look over tens of seconds, these elongated zones are persistent. And occasionally we do see a vesicle moving, but the majority of events are indeed elongated structures. Why is it that this kind of structure is favored? Could it be some kind of interaction with the actin cytoskeleton, which forms these linear structures close to the membrane, as was proposed and visualized by Akihiro Kasumi's lab? These corrals formed where the actin anchors things in the membrane might explain why the HA makes these elongated shapes. But to really test this question, we needed a technological advance. And so this was where multicolor was invented and immediately useful. And the one way of doing this is to take a dichroic in the detection path and split the light into separate channels, which are visualized on side-by-side regions of the camera. This is raw data with localization superimposed. And then we take an intensity ratio of the transmitted intensity over the reflected plus transmitted, which gives us a number between 0 and 1, which we call alpha. And the alpha value for one type of fluorophore is typically different from another type, and we can then separate them spectrally and get then even 3 or 4 things at once for 3 different fluorescent proteins. This allowed us to then visualize the actin with PAM cherry and the HA with dendra-2, both fluorescent proteins, and see the HA clusters close to the membrane where the cortical actin was enriched, thus supporting the idea that actin may play a role. We also found in other types of views, top-down views, for example, this same relationship also in different cell types, live and fixed cells. Seems to be a fairly robust finding. We also see that actin binding proteins co-localize with HA in a variety of structures. I would point out that some of these same actin binding proteins are found in purified flu virus. So this connection may not be coincidental. Yet, how can it work? HA has an ectodomain that sticks out of the cell, a transmembrane domain that's buried in the membrane, and a short, roughly 10 amino acid tail, which is the only part that sticks into the cell. How is it that this 10 amino acid long tail could bind to actin directly? There is no known actin binding between the tail and a monomer or an actin binding protein. And it's so small that it isn't even big enough to contain an actin binding domain. So how could this happen? How can this work? I got a clue from the fact that there are some highly conserved features in this cytoplasmic tail. We started to think, could this happen through a lipid rather than being a direct interaction? And the conservation in the tail is around specific cysteines, 3 of them, and those cysteines are acylated, so they have a lipid tail added, typically palmitate. And in addition, adjacent to these cysteines are arginines or lysines, which are positively charged. Thus, the thing that might interact could be something in the membrane that would interact with the acyl groups, and something which is negatively charged to have an attraction to these positive residues. And since HA is a trimer, if you have two positive charges, you really have a +6 charge per trimer and 9 acyl groups. So Nikki Kurthoy is a brilliant postdoc in the lab, suggested that this might be a way for PIP2 or phosphatidylinositol 4,5-bisphosphate to interact with the tail. Might be a good candidate. Has a very negative head group, and it's known to modulate a number of signaling pathways that have been implicated in influenza infection. It's also a known interactor with actin and actin binding proteins. We can also label it using a plexion homology domain and put a fluorescent protein on that, and we can visualize the PIP2 that's not bound to anything. Also, of the ABPs, the actin binding proteins found in flu, 14 of 16 are known to interact with PIP2. So this is pretty good indirect evidence. Super resolution gave us direct evidence. Labeling the PIP2 with the pH domain attached to PAMCate and labeling the HA with Dendra2, we see these regions of strong colocalization between HA and PIP2 at the nanoscale. We also did molecular dynamics simulations, especially during the beginning of the pandemic when we were all stuck at home, and we found that arginines in the tail, these positive regions, do feasibly interact with the negative charges in PIP2, and that the same thing seems to happen for the SARS-CoV-2 spike. So we decided to then do an all-atom simulation, having seen the coarse-grained simulation here suggesting feasibility. The all-atom simulation replicated that finding and made it more quantitative. And here's an example of an arginine from an HA trimer interacting with a PIP2 head group close to the plasma membrane. And this is in collaboration with Alex Sote, who's a molecular dynamics expert at NIH. So back to experiments now. If we mess with the tail, we should be able to disrupt the interaction. If we understand the interaction to be a charge-charge interaction and an acyl lipid tail interaction, that's what I'm proposing here. If we remove the acyl groups, that's May, and we change the positive charges to either negative or neutral, that's Hare, we don't really see alone a fundamental change in the attraction of PIP2 to HA. But when we do both, remove the charge and the acyl groups, we fundamentally change the interaction between HA and PIP2, suggesting that the tail is a major part of this interaction. So do we fully understand the mechanism? I would say there's still plenty left to think about, but what we think is happening at the moment is that we have free PIP2 in the plasma membrane. We have actin binding proteins and PIP2s interacting. The actin binding protein interacts with actin, and this would be without HA, kind of a normal cell membrane. The cell gets infected, the HA is trafficked to the plasma membrane, where the cytoplasmic tail of the HA can interact with PIP2, leading to a colocalization of HA with PIP2, as well as with actin binding proteins and actin. However, the HA may also be able to modulate PIP2 because it's binding what's available. That's changing the local concentration of PIP2, and that can modulate certain signaling pathways. So we're following up on this basically pretty good understanding, I think, but we would like to investigate further into signaling, and also, can we manipulate the PIP2 and disrupt the virus? Because of the conservation of that tail, this would be a good candidate for an antiviral therapy. So in collaboration with Professor Julie Goss at the University of Maine, who identified a compound called cetyl pyridinium chloride, which is an antimicrobial compound. It's basically a soap at its normal millimolar concentration. It's in all kinds of consumer products. She identified this as a putative molecule that could interact with the plasma membrane, has a positive charge, and could potentially screen the PIP2 away from other things that would bind it. And this turns out to be correct. If we look at MARCS, which is a PIP2 binding protein, normally it's associated with the plasma membrane. We add some CPC and some of the MARCS falls off. We add more CPC and more MARCS falls off, and the association with the plasma membrane decreases significantly. Thus, CPC seems to be able to steal some of the PIP2 or screen it from interacting with things that would like to interact with PIP2. What does this do to HA? Addition of CPC strongly disrupts HA clustering. And this is at micromolar concentrations that are well below the critical micelle concentration that one might suspect if it were acting as a soap. Also, the co-localization between HA and PIP2 decreases when CPC is applied. The HA cluster density drops significantly. And the same thing happens for the SARS-CoV-2 spike. So these are published in a couple of recent papers this year. We also have a story connecting the matrix protein, which is found inside the viral envelope and helps with assembly and budding, and is normally thought to interact with HA. We see it interacting. When we mutate the tail, which we think interacts with PIP2, the HA and M1 no longer interact. And when we deplete the PIP2 using CPC, the pH that's found near M1 decreases. The concentration of M1 in its own clusters decreases, and the colocalization with HA drops as well. So there seems to be an interaction between PIP2 and M1, which modulates the interaction between HA and M1, and can also be disrupted by this same drug. Also, I mentioned that SARS-CoV-2 spike contains the same features, the acylated cysteines and the positive charges, and it's actually to an even greater degree than HA. And when we imaged the spike together with pH, again, we see these regions of strong colocalization, very reminiscent of the HA-PIP2 interaction. Now, what I've talked about so far has been entirely in cell culture. And to do virus infections, you can do those in cell culture. But to get more physiological relevance, it's nice to have an animal model. And I'm lucky at University of Maine, we have an excellent zebrafish facility, people to help breed and grow and maintain zebrafish for various purposes. A really talented graduate student in the lab was the first to do localization microscopy in vivo in a live zebrafish, Kristen Gabor, which was published in PLOS One in 2015. And she looked at individual molecules of caveolin and was able to visualize structures in membranes up to 10 micrometers into a cell that was inside of the living zebrafish. More recently, we were able to extend this work to look at influenza because the zebrafish is also able to be infected by influenza. Its physiology is something that can be quantified as a function of days post-infection. Many of the zebrafish have significant morbidity at 5 days post-infection. And when we treat with CPC, however, they survive. And we are able to visualize HA clusters inside of those zebrafish because they're optically clear and the Casper mutant is fairly low background. So this offers an opportunity to do in vivo infection in an animal model, visualize the viral structures with super resolution, and do drug testing. And these zebrafish are able to reproduce and be genetically modified on a fairly short time scale so that we can do fairly high throughput testing. So that was also part of this toxicology and applied pharmacology paper. One last application I'll mention, muscular dystrophy is obviously a significant health problem, and one version of that disease can be modeled in the zebrafish as well. in order to make this work, we have to also be able to do super resolution of the muscle. And you can hopefully see that the confocal is able to see the T tubules, but the localization microscope sees them more distinctly. And when we quantify the cross sections of these tubules, in the confocal, they have a width which is about what you'd expect, the diffraction limited resolution of 300 or 400 nanometers. In the super resolution scope, those profiles narrow to 57 or so nanometers, demonstrating that the features are better resolved. And thus, if we need to study those muscle structures and compare disease and non-disease fish, we're able to do that with localization. Not to leave out the capability of multicolor as well in the zebrafish, which allows the visualization of intermolecular interactions that are typically very important in disease. So in summary, super resolution microscopy was developed in the late 2000s. I was very lucky to be involved in the early development of localization microscopy. And it was really because of my desire to study viruses that I pushed, and I had the need for it, and an immediate biological application once we got it to work. We were then able to discover that the clustering of HA depends on actin and actin binding proteins, which was a bit of a mystery because it wasn't clear how this could happen. We followed up advancing the technology to give multicolor and three-dimensional methods, and discovered that the phosphoinositide PIP2 plays a major role in HA's clustering, as well as HA's interactions with actin and actin binding proteins, and that interaction occurs through the cytoplasmic tail of HA, which is very, very highly conserved in certain ways. When the pandemic hit, we rushed to try to help, and a collaborator at NIH was able to make a FPALM compatible spike, which we could then image, and we saw also interacted with PIP2. We also have supporting information from molecular dynamics simulations that verify and actually give much better detail than you could ever achieve in a microscope, right? Individual positions of atoms. So these interactions between HA and PIP2, we think, explain the dependence of the virus on phosphoinositides, the interaction between HA and actin, and also offer a pharmacological attack that we can make on many viruses. I mean, it's actually surprising, but HIV has a known interaction with PIP2, Ebola also, hepatitis, and then HA and SARS-CoV-2, and there's a few other viruses I'm forgetting. But many of them use phosphoinositides to ride around the cell or assemble things. So, the conservation of this makes it hopefully something that can work on a variety of viruses and will continue to work even though the viruses are going to mutate. This cetylpyridinium chloride, which it's actually found in Colgate Total mouthwash, we found was able to disrupt HA and spike clustering and improve the survival of flu-infected zebrafish. There was actually some clinical trials to use this against COVID. And really, I think overall, this interaction between phosphoinositides and viruses is important and potentially powerful. So we're very committed to better understanding this. Hopefully the zebrafish can be a way to do that as well, since it allows us to test drugs pretty efficiently, as well as image what's going on. So let me acknowledge many hardworking students who've contributed to this. Current students, Komala and Mary, are the leaders of the muscular dystrophy work. Komala is also from Bangalore, which is pretty cool, and she's been developing a new super resolution version of FCS, which hopefully at some point I could talk to you more about. David and Brandon are both involved in the phosphoinositide work. David has been leading the all of the molecular dynamics work lately. And Nikki was the first to suggest we look at PIP2. And together with Jenny Lillyholm, they made some of the first observations of PIP2 in our systems. These folks were crucial in the development of FPALM and multicolor. Mike also developed a better multicolor method that individual molecular spectra can be imaged with. Kristen, I mentioned doing the in vivo developments, and then Prakash in the virus work, most recently with flu, phosphoinositides, and SARS-CoV-2 was absolutely crucial. Matt and Jackie also made important contributions, and Kashif developed that pH domain construct that we use to image PIP2. These are some of my collaborators, and you'll see Partha there too, who's visited the lab several times and has been a wonderful collaborator as well. And these are some of my funding sources. So thanks very much for your attention, and I would be happy to take any questions.
So thank you, Sam, for the enlightening talk and really focused and quite absorbing. Any questions that you people have, if you want to ask? Yeah.
So he's focusing on the zebrafish. Is this the same kind of experiment can be done in C. elegans also, sir?
Sam, did you understood the question?
Was a question about comparison of C. elegans and zebrafish?
Yeah, so yeah, same. Yeah. So can we do the same kind of experiments with C. elegans as well?
I think so. I haven't, I don't know as much about the fluorescence background levels of C. elegans. I think that's really, if that's a surmountable problem or is already good, then I think there's no reason localization couldn't work in C. elegans. The trouble with a lot of animals is that they have pigments, and the only reason we can do this in zebrafish is because of the Casper mutant, which gets rid of the pigment. And I'm not as familiar with the pigmentation of C. elegans, although I feel like there's not much. But that would be a crucial question to look at. What part of the spectrum has the least background? And then if you can choose your probes to match that spectral region, then it would probably work. But I guess the biological question needs to be tailored such that the animal isn't moving too much on the time scale of imaging, the structures of interest aren't moving too much, those kinds of things. But in principle, I think so.
Okay, anyone else? Sam, I have a few of them. So
please.
You talked about, yeah, I just want to know if this, the study that you have done that leads to clustering, does it translate to other viral diseases as well?
Well, that's a great question. Are you asking, does the methodology translate, or do the findings, the biological findings, translate?
Yeah, I'm just talking about the clustering part. So you showed.
Do other viral proteins cluster in the same way?
Right.
Well, yeah, the spike seems to, although assembly of the coronavirus is not at the plasma membrane. HIV GAG forms clusters in membranes. VSV G protein, I believe, clusters in membranes. So there are some other examples. Are they phosphoinositide dependent? I think the GAG is, although I'm not sure if anyone has done super res imaging with phosphoinositide modulation specifically, but there's that known binding, so I guess I would expect it would. Try to think of some other examples. I mean, many of the envelope viruses have non-uniform protein distributions. And since it's derived from cell membranes, it's likely to have the heterogeneity that cell membranes have, or more clustering than that. So I think there's some parallels.
We have a question from one of the participant, I mean candidates. Arvind, do you want to repeat your question?
Yes, sir. So actually, like when we consider fusion proteins like dendrite to HA, its size will be greater than viral protein like HA. So this extra size, will it affect the study or will it play any role in imaging?
Yeah, that's a great question. I think it probably does. I mean, the function of these viral fusion proteins is pretty highly evolved and selected, you know. We've had to be pretty careful to put the fluorophore in places we think will not interfere with trafficking, will not interfere with cleavage, will not interfere with exposure of the fusion peptide. And so our fluorophores are typically on the end of the cytoplasmic tail or on a loop on the ectodomain that we think does not affect, is not close to the fusion peptide. But so I mean, you're talking about 120,000 molecular weight HA and a 28 to 30,000 molecular weight fluorescent protein. We were pretty nervous about this, and so we tried changing the location of the labeling site, changing the label itself, using antibodies to label. So we're labeling native protein. And other groups have used organic dyes to label a specific location within the protein, and those are much smaller. I would argue that these results are generally in agreement with each other. And so we feel, oh, and the immunogold I also compared. So, you know, the fact that we're seeing similar patterns for all these different ways of labeling is reassuring, although it doesn't, you know, it doesn't eliminate the chance that something could be altered. So that's a great question.
Yeah, Sam, one more question is how helpful is molecular dynamics?
I love it. I mean, I've been seduced maybe, or I feel it's a great complement to experiments. It's not a substitute to me. It's not a substitute for experiments, right? It's still not the real world necessarily, but it keeps getting better. Computer resources keep expanding, and the ability to make these two work together is awesome. I mean, you do the experiment, you do the simulation, you see how they can test each other, and then the net is stronger than the sum of the parts. So I think it's a great thing to combine with experiments.
Right. Another thing that comes to mind is basically what could be the good drug candidates, and what do you think? What is the stage that it should act at the entry inside, inside the cell, or what is the stage? Do you think the drug candidate is better, best placed?
I mean, any stage that's important can be attacked, and some of the existing inhibitors, target the proton channel or the neuraminidase, so those work. I think probably the best candidates are the ones that use invariant features of the protein. And so this could happen at any stage in the life cycle, but the crucial thing is that the virus not be able to mutate and avoid the effect of the drug. Because sooner, as soon as the thing is used widely, the virus has a chance to mutate, and any form which is better surviving will be grown better, will be selected. So I'm not answering your question because I think it could be any of the stages. but the key thing is to have something which is invariant, if possible, something universal.
Yeah.
We like working on assembly and fusion because those are things that involve memory and organization, and so we can visualize the effects with our techniques. But you know, Xiaowei Zhuang has done a great deal of work in Flu on other stages, the binding and entry stage in particular, and has had some great success. So
yeah. Finally, Sam, PIP2 interaction with PIP2 seems to be central to many. I mean, at least you talked about a couple of diseases.
yeah.
So do you have any? Do you have more information about this as far as other viral diseases are concerned?
Yeah, I mean, the literature is immense on roles of viruses, both phosphoinositides in viral life cycles and in infection. I mean, the and the variety of phosphoinositides is also quite broad, right? You have PIP2, but that's, you know, I'm talking about the 4,5 PIP2, but you could have a 3,4 or 3,5 PIP2. You can have a 3,4,5 PIP3. You have all the individual single phosphate PIPs like PI 3P, 4P, 5P. And all of those seem to play physiological roles in the cell. Probably all of those are exploited by viruses in some way. And the number of functions controlled by PIPs is immense. So, you know, it's hard to wrap one's mind around all the literature that's out there. There's a great deal. You know, the PI3P family has been implicated quite a bit in other viruses, SARS-CoV-2, for example, but many others. So, I mean, that's just viruses. And then, you know, these relate to inflammation, mitochondrial function, calcium signaling, neurotransmission, right? These PIPs are all over the place. So viruses that attack specific organs or specific functions may be hijacking different sets of PIPs. So I really feel there's a lot there. It's hard to really stay on top of it.
Okay. I don't see. Oh, there is one more question that comes up. Sorry,
Please,
Sam.
please,
Yeah.
that's great. I love the questions.
Sir, you told mutation of cells of influenza leads to the detection of the infection. Means due to the cluster formation, you detect the influenza, right? So in cancer also mutation takes place. From that, can you detect cancer?
Martha, do you think you could repeat what you understand?
Sure. Influenza virus leads to the mutation of cells, so clusters are formed, right? From that, you are detecting that it's a virus. So in cancer also mutation takes place. Can you detect cancer using them?
Oh, can you detect cancer using some of these same strategies?
Yeah.
It's a good question. I think a specific kind of cancer could certainly be tested. The changes in the cell structure, signaling pathways, trafficking of things, the growth, control of growth or lack thereof. I think all of those things could be tested with these kinds of methods. But each kind of cancer is distinct. And so general mechanism might be maybe centered around growth and response to neighboring cells, in which case you might want to consider a more realistic cell environment like a tissue or spheroid or something that's taken out of, you know, ex vivo, grown in culture or something. And then using the same methods to test the discerning features of that type of cancer. So in principle, yes, but it would, in detail, it would require knowing how that particular type of cancer is identified.
Thank you, sir.
Not sure. Did I answer your question?
Yeah. Yes, sir.
I believe, Sam, that henceforth super resolution microscopy is going to be central to disease biology and all kinds of biology.
I hope so.
Yeah, it looks like, because I mean, single molecule could be the best that we can do. Yeah. Anyway, I don't. One more question is there.
Please, that's great.
Protein of interest use the protein of interest can be changed the photophysical properties of the fluorophore, whether it can be changed. Protein of interest which we have used with the fluorophore, whether it can change the photophysical properties of the fluorophore, whether it will mask the chromophore or something like that, whether it can happen.
I heard some. Parthik, do you think you can?
Yeah.
Protein of interest which we have fused to the fluorophore, whether it will change that photophysical properties of that fluorophore, whether it will mask the fluorescence or some faulting can affect the fluorescence or something, or spectral change can happen, something like
Yes.
that. Is it possible?
Yes, excellent question. I mean, I worry about these things, and I also hope for them sometimes. I mean, the effect of something attached to the fluorophore, if all you want is the location of the fluorophore, and you collect as much light as you can from it, then the position may be fine. But if you're looking at the emission of the fluorophore, it is certainly dependent on the environment and things that are bound. And the pH can affect the emission, the temperature, the oxygen, the redox state, the membrane potential. I mean, all of these green fluorescent protein versions that have been designed to be responsive to the environment show ways that GFP itself may be responsive to the environment or to things bound. So absolutely, the GFP or the proteins we use, which are derived from GFP, are sensitive to the environment and to attachment of things. And depending on what you're interested in, that can be a problem, or it can be a way of measuring that quantity using the fluorescence data that's encoded. So yeah, I love that question.
Okay, I'll see if there are more. I don't see any more. So Sam, I guess thanks a lot for the wonderful talk, and we all
My
really
pleasure.
love to see you here, and hope to see you in future as well, because you are so busy to really take your time out.
Oh,
And, yeah,
my pleasure.
yeah.
My pleasure.
Thanks a lot, Sam. I think we are kind of done with the. I think no more questions are there. So, right.
Okay.
Yeah.
Thanks, Partha.
Yeah.
Thanks, everyone.
Yeah.
Have a good day.
Yes. Stop.